I'd like to introduce our next speaker. It's Dr. Brian Bahder. Brian received his bachelor's at University of Delaware and his master's at University of Florida, and then he received his PhD with Dr. Doug Walsh at Washington State University, where he worked on the epidemiology of grapevine leaf roll-associated viruses in Washington State vineyards. He joined Sudi and myself here at UC Davis in 2014 on our project trying to identify a vector species that might be responsible for transmitting the virus. So I'll sort of share my screen here with Brian. And
okay. Thank you, Frank. And can everybody hear me?
Yes.
Okay, great. Okay, so I'd like to thank everybody for being here today to listen to my presentation. And I'm going to be giving an update on our search for a vector of grapevine red blotch-associated virus. Oops. There we go. Okay. To kind of start off with, I wanted to go over my transmission assays that I've done with Erythroneura leafhoppers. Erythroneura zigzag is the Virginia creeper leafhopper that was implicated as a vector. And when I started my research, that was kind of the baseline that I wanted to look at it, see if I could replicate it, as well as look at some of the other common Erythroneura leafhoppers that we have in California and on the West Coast. And so far, my initial assays that I did pretty soon after I got here, they're at about a year and a half post-inoculation, and none of those plants are testing positive for red blotch. So I decided to do a more intensive set of experiments with these species by exposing them to more insects and for a longer period of time. And this was, it took me a little time to get the colonies established so that I could use more individuals to work with. But I'm at a 4-month post-inoculation test with these experiments, and so far I haven't achieved any successful transmission with any of these species. Now, I wanted to show some of our data from the field that kind of gives us a clue that the virus is in fact spreading in vineyards. Some of these maps look very similar to Von Walton's maps, as well as Keith Perry's maps on the distribution of infected vines within the vineyard. Now, these maps here represent one of the vineyard plots at the Oakville Research Station, where we have yearly data documenting increase in incidence based on PCR data. And you can see from this, from 2011 to 2014, there was an increase in grapevine red blotch infected vines. So this was kind of ground zero for our vector search. But since I got here, we've started investigating additional sites, and this one here is located in Amador County. And this is one that we just started looking at last year, so we don't have yearly data with PCR confirmation of newly infected vines. This is based off of symptoms and a subsample of PCR positive vines. But you can see just from the distribution, like you saw in Vaughn and Keith's maps, you get a distinct clustering and edge effect, which is pretty indicative of a vector. And we're going to be following up with this vineyard in the next year or so to document any increase in incidence. As well, this is another vineyard in Santa Barbara where for about 70 years it was healthy, clean, no red blotch symptoms on any of the vines. And then in the last few years they brought in new material from a nursery, planted it, and you can see that all of a sudden you get a big cluster on the edge of the vineyard, which is, again, indicative of a vector. Now, one of my main research projects has been evaluating vineyards where we either see spread or suspect spread versus vineyards where there is no apparent spread occurring. And I've been surveying for insects and comparing the species that I'm finding at each of these vineyards. And from that, I've been able to come up with a short list of species that I believe are likely going to be the vectors of this virus. Now, to kind of go along with this, I did an analysis of the coat protein amino acid residues to look at the evolutionary relationship of red blotch versus other Gemini viruses. And this tree here shows that our red blotch virus, in terms of the coat protein, comes out very, very close to another Gemini virus, the tomato pseudo curly top virus, and this Gemini virus is transmitted by a membracid. So between that and all the other Gemini viruses transmitted either by a leafhopper or whitefly, this kind of gave me the idea that the vector we're looking for is likely going to be a hemipteran insect, and I suspected that it was within the Alkenaranca, that includes membracids, cicadelids, insects within that taxonomic grouping. So from that, my list matched up with that pretty well. The 5 species that I suspected from these vineyards were a membracid, 2 species of leafhopper, and a jumping plant lice, as well as a plant hopper, a succeed. And I had done quite a few experiments with each of these species, and I have lots of assays going in the greenhouse at this point, and I'm still testing them at their different post-inoculation intervals. And from this short list, these are the preliminary results of the transmission assays I've conducted. And out of these assays, the only one where I have achieved transmission within the greenhouse is from our membracid. And I'm going to show you guys the species here in a moment, but I also wanted to include the other species showing that there was negative data from these other highly likely candidates. The 2 leafhoppers were the sharp-nosed leafhopper, Scaphitopius cutis, and the second leafhopper was a Fibriella florii. I think I saw that species in Vaughn's presentation as well. The succeed is a Melanoliera species. I haven't identified it to species. They're kind of difficult. And the psyllid is a potato psyllid. So to test for red blotch in my recipient plants, we've been using digital PCR, a chip-based system similar to Sudarshan's droplet digital PCR. And this technology is quite a bit more sensitive than quantitative PCR. And to test my recipient plants, since I had so many transmission assays, I did cohorts of 15 plants, and I extracted the DNA from all 15 plants together, and I tested it with digital PCR. And because of the sensitivity, I could still theoretically detect the virus in those pooled samples. And these are the results of my digital PCR assays. Now you can see here on the left, this image of a digital PCR chip shows no amplification at all. The yellow wells are no fluorescence, and the white ones are wells that were deleted by the software because they didn't meet the quality threshold. And I didn't start detecting red blotch in these pooled samples until 3 months post-inoculation. And even then, it's an extremely low concentration. There's about 21 wells out of about 18,000 that tested positive for red blotch. but you can see at 4 months you start to see an increase in positive reactions through the digital PCR assays. And then at 5 months, you start to see quite a few more. So at 5 months, after I had 3 months of consecutive increase in viral titers from these cohorts of plants, I extracted DNA from each plant individually and ran standard PCR assays on each of these experimental plants to see if I could detect it and get it sequenced from individual plants. And from that, I was able to get a faint reaction on 3 of the 15 assays I had conducted with this 3-cornered alfalfa tree hopper. The species name is Spicistulus festinus. And this species, based on my assays in the greenhouse, was able to transmit the virus to 3 plants out of my 15 experiments. And this is based on individual insects exposed to a source plant for 48 hours and then moved to a recipient plant for 48 hours. Now, we just got the sequence data from these individual plants and confirmed that it was in fact red blotch and it matched our source plant. And you know, we're getting a paper ready to kind of touch on these results and go over the transmission of red blotch under greenhouse conditions with this species. And you can see here in the image I have the actual insect. The male is on the left. It has kind of the red highlight on the pronotum, whereas the female is slightly larger and has a white ridge there. And on the right is kind of characteristic damage that you see in vineyards here in California where there's girdling, and that's due to the feeding behavior of this tree hopper. And so the digital PCR combined with our standard PCR and sequence data, we were able to demonstrate this species' ability to transmit the virus under greenhouse conditions. Now, it's important to note we're not specifically saying this is the only vector out there, or if it's and if it's not the only vector, if it's responsible for most of the spread that we see in California. You know, we got to follow up on the species and look at the true ability and of this insect to vector it in a field setting as opposed to greenhouse conditions. So based on these results, it doesn't look like any of the Erythroneura species are likely vectors, but we are still testing these plants over time to see if we can detect the virus in them. And as well as these other species that I thought were likely candidates, we're still testing them over time because we don't want to just assume that it's only one species transmitting the virus. And of these candidate species, we can detect infections in plants much faster due to the sensitivity of digital PCR. And the tree hopper that we were able to obtain transmission with under greenhouse conditions is kind of an interesting insect because grapes are not the preferred host of this species. They prefer legumes and grasses and things of that nature, which you tend to see on the edge of vineyards near riparian habitat, which kind of fits the mold with the distribution of virus we see in certain vineyards. And it's also restricted more to the southern U.S., and it's native to North America, which kind of fits with the mold of only seeing spread in California and in Oregon, whereas from my understanding in Washington, New York, and other grape-growing regions in North America, as well as internationally, spread isn't seen. That kind of fits the mold with this species being more restricted in geographic range that could attribute to variation in spread that we see from place to place. So that I'd like to thank our funding sources and our collaborators, and I will turn it back to Frank. That's great.
Okay, well, thank you, Brian. I appreciate the presentation, and Brian will be on the question and answer session immediately after this as well.